This lecture is on cardiac physiology, part 3. So far, we've talked about the cardiac action potential and the autorhythmic cell action potential and how that travels through the heart. We've talked about the intrinsic conduction system and valves and chambers, EKG, and the flow of blood through the heart. So now we're going to try to pull that all together and talk about the events that occurred during one complete heartbeat. That's called the cardiac cycle. Systole is a period when the atria or the ventricles are contracting. Diastole is a period when the atria or the ventricles are relaxing. So if you think about it, there should be 4 steps to the cardiac cycle: atrial systole, atrial diastole, ventricular systole, and then ventricular diastole. The cardiac cycle takes about 0.8 seconds. So for one heartbeat, one cardiac cycle, it's about 0.8 seconds. But it turns out that some scientists have put the cardiac cycle into 3 steps or phases. Now in your textbook, the Martini textbook, they break it into 5 phases. But 3 phases is easier to remember, so we're going to go with that. Now this is a busy picture, and of course one picture can tell 1,000 words, or hold 1,000 words. But in this one, I think what you're going to come to realize is that you already know all of these things. You know what's going on in the heart, because we've already talked about them. And so this is just about putting it together. At the bottom of the screen, we can see phase 1, 2A, 2B, and 3. And then again, 1, 2A, 2B, and 3, and over again. There actually is a name for each phase: stage ventricular filling, ventricular systole, early diastole, and then within each phase there are descriptors, very simple descriptors that help you remember what's going on. Here we have pictures, which I think is the most helpful thing. And then in this part here, you can see whether the valves are opened or closed. These are the semilunar valves, the aortic semilunar and the pulmonary semilunar, and then whether the atrioventricular valves are open or closed. In this graph, it's showing ventricular volume in milliliters. This graph is showing you the pressure in different chambers and blood vessels. So the green line is the pressure in the aorta, the dark blue line is the pressure in the left ventricle, the purple line in the left atrium. And then at the top here, they're showing you the ECG or EKG. We've talked about this, and then also heart sounds, which we haven't talked about. So I'm really not going to talk about heart sounds, because I think that you will probably get a better explanation in the program that you go in the health field. But briefly, the sounds that you hear when you're listening to someone's heart are the closing of the semilunar valves. So let's start with this first phase, phase 1. And in terms of what's happening, well, it tells you the ventricles are filling and the atria contract in that order. So here, if we start with this one, you'll see that in this phase, the blood is returning to the heart and it's filling up the atria. When the atria get enough blood in them, they push through by the valves, the AV valves, and they start to fill the ventricles. The ventricles are filling. Now, the atria then have to contract in order to squeeze out the last drops of blood into the ventricles. If we look at this picture here, you can look and see that the atrioventricular valves, the tricuspid valve on the right side of the heart, the bicuspid on the left, are open, because they have to be open to let the blood flow. The semilunar valves are closed, and so that's what it's telling you up here. Semilunar valves closed, AV valves open. What's happening to the volume? Well, what do you think would be happening? It's slowly increasing as the blood returns to the heart. This little bump here occurs after the atria contract. So there's a little bump in volume because they're squeezing out the last bits of blood. If we look at the pressure graph, you'll notice the green line for the aorta, the pressure is going down. Now pressure is often a kind of a measure of how much blood is there, because the force of the blood on the walls of the vessel is what's causing the pressure. The reason it's going down in the aorta is that just before this whole phase here, the ventricles contracted and the blood hit the aorta, and now it's slowly leaving it, and as it does, the pressure goes down. What's happening in the atrium is that there's a little bump in pressure here when the atria contract. What's going on with the EKG? Well, for a long time, nothing, and then you see the P wave. Now remember, right after the P wave is where the atria contract, right? So the P wave is depolarization of the atria, and right after it, they contract, and that's what's happening in phase 1. So if we go to this next slide here, let's just summarize this. What's happening in phase 1? Well, the ventricles are filling, and the atria contract. Okay, what valves are open? The atrioventricular valves. Which ones are closed? The semilunar valves. Okay, let's go back to this picture again, and now we're going to move on to phase 2. And you can see that there are actually 2 steps in this phase. There's the isovolumetric contraction phase and the ventricular ejection phase. So let's start with this one first. And what does that word mean, isovolumetric? Well, iso means constant. And so what this word means literally is constant volume. Now, if we look at this picture up here, this is isolating this phase right here, isovolumetric contraction phase, isovolumetric contraction phase. So what's going on here is you'll notice, first of all, which valves are closed. So if you said all of them, you're correct. The semilunar valves are closed and the AV valves are closed. So which ones are open? None of them. And what's happening now, well, this is a split second in time when the valves are closed and the volume of blood in both ventricles is constant. Okay, so this is a split second in time when the volume of blood in both ventricles is constant. Okay, now the reason it's constant is it's not going anywhere. All the valves are closed. I'm just steering a little bit away of saying that the volumes are the same, because the volumes in both ventricles are pretty close, but they're not exactly the same. But in this case, they are constant. Now, if we go back to this picture, and here we are, isovolumetric contraction phase. If we move up here, you can see both types of valves are closed. And so we're following this light salmon color up here. You'll notice that the volume of blood in the ventricle is pretty high. In fact, it's the highest it's ever going to be in the whole cardiac cycle. And so this is called the end diastolic volume. Now at this point, the end diastolic volume is the maximum amount of blood that the ventricles will ever have in the whole cardiac cycle. Okay, so let's write that down. End diastolic volume is the maximum amount of blood that the ventricles will ever have. At least in the cardiac cycle. And again, if we go up here and take a look at this, and I said to you, what's the volume like, high or low? You would say, oh, well, it's high, because it's as high as it can ever be. So we'll give that an up arrow. What about pressure? Well, if volume of blood is high, the pressure is high. Now, the next thing that's going to happen is that the ventricles are going to eject the blood. This is called the ventricular ejection phase. And again, we're going to go back to this picture here, and now we are up here in this salmon-colored, light salmon-colored, and you'll notice that on the EKG, we're at the end of the QRS wave. So remember that right after QRS, what happens is contraction of the ventricles, and that's what's going on here. Okay, so in this phase, the ventricular ejection phase, the ventricles contract and the blood is forced out. Okay. Let's go back to the picture now again, and we're going to go to the last phase, phase 3, which is called isovolumetric relaxation phase. So there we are with the isovolumetric again. Now, if I said to you which valves are closed, you can clearly see they're all closed. And if I said to you, what does isovolumetric mean? You might say, oh, it means constant volume. So you'll notice that the ventricles had just ejected the blood, but there's still some blood left in both ventricles. So it doesn't eject all the blood. But we can say that the volume of blood in both ventricles here is constant. And so if we look at the volume here, you'll notice that in phase 3, it's really low. In fact, it's the lowest it will ever be in the whole cardiac cycle. So they call this end systolic volume. End systolic volume. So again, in phase 3, what is happening is the volume of blood in both ventricles is constant again. In terms of the volume, however, it's low. And if the volume is low, so is the pressure. What valves are open? Well, you can see from this picture right here, none. What valves are closed? All of them. And what's going on is we have reached the end systolic volume, and this is when the ventricles contain the least amount of blood that they will have in the entire cardiac cycle. Okay, so let's go back to this picture one more time here, and we're in phase 3. Okay, you'll notice that in terms of the EKG, we're right at the end of the T wave, and what happens, remember, the T wave is repolarization of the ventricles. But to go back here, here is the end systolic volume, the least amount of blood the ventricles will have. Here's the end diastolic, the most amount. So if I wanted to know how much blood was actually pumped out by the ventricles, I could take the maximum amount here, end diastolic, and subtract it from what's left over and still in the ventricles, the ESV. And in fact, we can do that. So if we take the end diastolic volume, which is the maximum amount of blood that the ventricles will ever have, and we subtract it from the end systolic volume, the minimum amount of blood, we get something called the stroke volume. The stroke volume is the amount of blood pumped out of each ventricle in one heartbeat. And this is going to be something that we talk about a little bit later, stroke volume, so I won't write it down here. Okay, so all of this there's a lot of stuff going on here in the heart during one heartbeat. But before you lose sight of the forest for the trees, remember that the reason blood is flowing through the heart is because of pressure changes. Those pressure changes are due to the cardiac muscle contracting and relaxing in different parts of the heart. And blood, like all liquids, is going to flow from a high to a low pressure. So that's the bottom line in terms of the heart. Now, this is probably a more practical term here, something called cardiac output. But just to go back to what we mentioned before, stroke volume. So remember we said stroke volume is the amount of blood pumped out of each ventricle in a heartbeat. And we can figure this out by taking the end diastolic volume, which is the maximum amount of blood the ventricles ever have, and subtracting it from the end systolic volume, which is the minimum amount of blood, what's left over after the ventricles contract. So this is called stroke volume. If you want to know the amount of blood pumped out by each ventricle in a minute, that's called the cardiac output, or CO for short. So cardiac output is a common measure of the efficiency of your heart. How much blood does each ventricle pump out in a minute? And you can figure this out if you have some information. So let's take an example here, and I'm going to tell you that this patient has a heart rate of about 75 beats per minute. Okay, so the person is at rest right now. And their stroke volume is about 70 milliliters per beat. Okay, so that means that in every heartbeat, one ventricle pumps out 70 milliliters of blood. And what I want to know is, what is their cardiac output? So of course, you're going to just take this formula. Heart rate, so cardiac output equals heart rate, 75 beats per minute, times stroke volume, 70 mils per beat. And if we do this, the beats will cancel out because they're in the numerator and denominator, and we end up with 5,250 milliliters per minute. This is the cardiac output. Now let's change it to liters. So there's 1,000 milliliters in a liter, so we need to move the decimal point 1, 2, 3. So this is the same as about 5.3 liters per minute. That's the cardiac output of a typical person at rest. 5 liters. How many liters of blood is there in a typical human? Well, somewhere between 4 to 5 liters in a female, 5 to 6 in a male. So what this means is that in 1 minute, your heart can pump out your entire blood volume. So it can circulate your blood volume through your entire body in 1 minute. That is a very efficient pump. And so again, cardiac output is a good measure of the efficiency of your heart as a pump. Cardiac reserve is another term that people sometimes use because cardiac output can vary. And this is basically the difference between your maximum cardiac output. So that's the maximum cardiac output is when you're exercising to the max, and your resting cardiac output when you're at rest. Okay? So cardiac reserve, again, is kind of like that, the extra amount of blood that you can pump out if you really had to. The difference between your max cardiac output and your resting. So for the average person, the cardiac reserve is around 4 to 5 times the resting cardiac output. 4 to 5 times. So remember we said it's about 5 liters. So the difference between your resting and your max is 4 to 5 times. So instead of pumping out 5 liters of blood, you would pump out 5 times 4 is 20, or 5 times 5 is 25. 20 to 25 liters of blood per minute. That's incredible. But for an athlete, the cardiac output, cardiac reserve, is even greater. It's 6 to 7 times cardiac rest. And that's pretty amazing. Now remember, athletes exercise for a living. So you might say, I want to have a bigger cardiac output. How can I get it? Well, remember, cardiac output equals heart rate times stroke volume. So by increasing either of these, you're going to increase the cardiac output. Well, heart rate changes throughout the day, but for most people, stroke volume is fairly constant. So you might focus on that and say, well, what affects stroke volume? A scientist named Dr. Frank Starling studied this issue, and he found that the most important thing that affects your stroke volume is something called preload. Preload is the degree of stretch of your cardiac muscles before they contract. So how stretched is cardiac muscle before it contracts? And the easiest way to show you this is with a sarcomere. So we have to remember back to AMP1 and skeletal muscles. So sarcomeres exist within cardiac muscles too. And remember, this is the structural unit of muscle. And we have the thin filament here, here, and here. And here is the thick filament, right? Thick filament with the myosin heads. And here we have the Z discs. So when this sarcomere contracts, the thin filaments are going to move inward like this. and they're going to move inward until they overlap. So the distance that this sarcomere is going to contract is this distance right here. We're going to call that x, okay? So that's how much it contracts, and the more it contracts, the more force it produces, the greater the preload. But let's say it's an athlete now. We're going to look at their cardiac muscle sarcomeres, and they're going to look something like this. So when they're at rest, this is what their sarcomeres look like. They're a lot more stretched. And so when this sarcomere contracts, it's going to contract over this distance. Now, if this distance up here is x, then this distance down here might be 2x, just eyeballing it. So that means that the sarcomere of an athlete is going to contract 2 times longer than that of an average person and is going to produce 2 times more force. And that's what preload is. It's about how stretched your cardiac muscles are before the muscle contracts. The more stretched, the more contraction, the more force. And you can see that in this picture right here. All right, so here's the typical heart where there's a solid black line, but then this part here with the dotted line is showing you the stretched myocardium, which means that when it contracts over a greater distance, more force, more blood pumped out. So now you know, well, if you want to increase your cardiac output, you should increase your preload. How do I do that? Well, basically anything that increases the speed or the volume of the blood returning to the heart is going to stretch the cardiac muscle. So think about it this way. If the speed with which the blood returns, if it's faster, as the blood slams into the ventricle, it's going to stretch it a little bit. And if this happens over and over and over and over again for days and months and maybe years, that stretch is going to stay stretched. Also, if the ventricles are able to fill more completely before they contract, that's going to stretch them. Now again, this isn't a one-time thing. This has to be constant over a period of time, but the ventricles will stretch. So what can you do that increases the volume or the speed of the return of blood? Well, one thing that comes to mind is exercise. When you exercise, you increase the rate of return of blood to your heart. And if you do this on a regular basis, you can actually stretch your myocardium a little and increase your preload, which means you'll increase your cardiac output. Your heart will be more efficient as a pump. How much exercise? As little as 30 minutes 3 times a week can increase your preload. What else? Well, remember we said you should increase the volume. Well, in order to do that, you want to lower your heart rate. If you lower your heart rate, it gives the ventricles more time to fill before they have to contract, and that will stretch them. So think of things like yoga or meditation. On the other hand, what kind of things decrease stroke volume? Well, anything that decreases the volume or speed of blood return. So an example of that might be blood loss. And of course, this is not a long-term thing, hopefully, but it can decrease your stroke volume. A rapid heart rate, and in some people, the heart rate is elevated. We're talking about greater than 160 beats per minute. So this is going to decrease your end diastolic volume, decrease the amount of blood remaining in the heart, and it's also going to decrease your preload. Okay, so there are people that have chronic rapid heart rate. Some people, especially diabetics, can have something called acidosis when there's too much hydrogen ions in their blood, and this can also decrease the volume and speed of return. And then there's something called afterload. Afterload is the force the ventricles have to overcome to push open the aortic and pulmonary semilunar valves. So it's the force the ventricles have to overcome to push open the semilunar valves. Now, in most people, this isn't an issue, but in people who have chronic high blood pressure, this is an issue, and it becomes harder to push the blood out because of high blood pressure in the aorta. And so their stroke volume can significantly decrease, as well as their preload. Another example would be alcoholics. In people who are alcoholics, the myocardium is damaged. And when it's damaged, it becomes flabby. The cardiac muscle cells die, they get replaced with connective tissue, and even though the ventricles are contracting, they're contracting with less force. And that's going to decrease your preload. Now, of course, if this is the situation for this particular patient, remember cardiac output equals heart rate times stroke volume. If your stroke volume drops, your body is going to try to maintain homeostasis. It wants to make sure the cardiac output is still sufficient. So if this goes down, the only way to keep this the same is to increase your heart rate. And this is something that happens in people who are alcoholics. As they destroy the myocardium and their stroke volume and preload drop, their heart rate goes up. But of course, that isn't good, because that doesn't allow the ventricles to fill completely. So it's kind of a vicious cycle. So again, in order to improve your cardiac output and the efficiency of your heart, you want to increase your preload. And exercise and finding ways to decrease your heart rate is a good way to start. But in this formula, cardiac output equals heart rate times stroke volume, we do have heart rate as an issue. Now, really, it's stroke volume that probably controls cardiac output in most people. But this is a good opportunity to just talk about how heart rate is affected by your nervous system. Now, remember, we said that the heart does not need the nervous system to initiate an action potential. However, the heart is still innervated by the sympathetic and the parasympathetic nervous system. It's not initiating a heart rate, an action potential, but it is moderating the speed with which the heart beats. So the parasympathetic, remember, this is the resting and digesting branch of the nervous system, and this is going to decrease your heart rate. The sympathetic, remember, the nickname for this is the fight or flight division, and this is going to increase your heart rate and ultimately cardiac output. There are other things that affect your heart rate, chemicals. So we have hormones, ions, and toxic chemicals. So for a couple of examples, if we look at epinephrine and norepinephrine, remember that these are hormones secreted by the adrenal medulla. Okay? Epinephrine and norepinephrine are going to increase your heart rate. They also increase the contractility, and if they increase the heart rate, they're going to increase the cardiac output. They do this by opening calcium channels in cardiac muscle via a second messenger system. Another hormone involved in this is thyroid hormone. Thyroid hormone, remember, is a general metabolic hormone released by the thyroid gland, and it is also going to increase your heart rate. So, again, people with hyperthyroidism have something called tachycardia, which is an elevated heart rate, which makes sense because they make too much thyroid hormone. And lastly, we have glucagon, another hormone made by the alpha cells in the pancreas, and this also increases your heart rate. Okay, what about ions? Well, we've got potassium and calcium. Potassium is going to decrease your heart rate by hyperpolarizing the membrane. Now remember, for the typical action potential, you have depolarization and then repolarization. So if the muscle cell starts out at minus 90 millivolts at rest, it depolarizes to plus 30, and then it repolarizes down to minus 90. But if there's too much calcium, it will repolarize below minus 90, maybe minus 110. And that makes it harder for it to depolarize. So too much potassium, called hyperkalemia, is very dangerous. And it essentially decreases your heart rate because it makes it harder for depolarization to occur and an action potential. Calcium is another ion that gets transported into cardiac muscle cells. It is not common, but excessive amounts of calcium prolongs the action potential. And it does that because it prolongs the plateau. So the longer the plateau, the longer the action potential, which means the slower the muscle contracts, the lower the heart rate. Deficiencies in calcium cause a shorter action potential for the same reason. It shortens the plateau, meaning the action potential is shorter, and so is the heart rate is quicker. And again, these aren't very common. The major effect of calcium on the heart is on contractility. All right, so since I'm a toxicologist, I have to mention some toxins, and I am just going to mention a couple. Digitalis is the first one, and Digitalis comes from the plant called foxglove, which you can see on the right. Foxglove is a native plant to New England, and I have lots of this in my garden. And it was discovered long ago that the flowers, the stem, the leaves of foxglove contain a chemical called digitalis. What this chemical does is it increases the contractility of the heart. And it does that by increasing the amount of calcium within the cell. Now remember, usually the more calcium, the more force. So if you have the right amount of digitalis, you can increase the contractility of the heart. At the same time, it decreases the heart rate because it stimulates the vagus nerve. Now remember, the vagus nerve is part of the parasympathetic nervous system, part of the resting and digesting. So physicians figured that this would be a great drug for people who had heart attack, had damage to the myocardium, because this is exactly what you want to happen. You want to increase the force with which the muscle contracts, because you've lost a lot of myocardium. But you also want to lower the heart rate. And so digitalis is a great drug for that. Nowadays, it's made in the lab, but the therapeutic dose is very close to the lethal dose. And what that means is the amount of the drug that will help you is not that much less than the amount of the drug that will kill you. So you have to be very careful with this particular medication, and of course this plant is very toxic to humans and animals. Ethanol is another example of a drug, and it depends on its use. So there can be short-term use of alcohol or long-term. Short-term use of alcohol decreases the heart rate because it depresses the central nervous system, and then it goes back to normal. But if there is long-term chronic abuse of alcohol, it is going to increase your heart rate. And the reason it does this is because it damages the myocardium. Now remember the formula, cardiac output equals heart rate times stroke volume. So as the myocardium is damaged, the stroke volume goes down. And as your body tries to maintain homeostasis and keep up that cardiac output, the only thing it can do is raise the heart rate. And that's what happens. So long-term abuse increases the heart rate. Now these other things at the bottom, the scorpion here and the cobra, these are not found in New England. However, I thought I'd mention them anyways because it's kind of interesting. The cobra secretes a toxin that is a cardiac toxin. It disrupts the cardiac muscle cell's plasma membrane. So it damages the plasma membrane, which kills the cell and basically can put you in systolic arrest. And so that's from the cobra. The scorpion, most of the scorpions in the US are not that toxic, at least to adults, but in other warmer parts of the world they are. And a lot of these release a toxin that slows the closing of the sodium channels and slows the opening of the potassium channels. So remember the action potential in a cardiac muscle cell. Here's where the sodium channel comes in. And if you slow its closing and you prevent the opening of the potassium channels, it can prevent the action potential. And so the heart can go into fibrillation, and which is how this works. One last thing I'll say about heart rate is it varies with age. So in very young newborns, their normal heart rate is about 120 beats per minute. But as you get older, that slows down. In women tend to have a slightly higher heart rate than men, somewhere between 78 to 82 beats per minute. Men's heart rate is a little slower, 70 to 72 beats per minute. And as you get older, your heart rate increases.